Before I'm going to come back to this slide. Before we get into that, I want to just give a little bit of background. How many people are familiar with the physical sample curation cluster already? Yeah, good number of folks. For those of you that aren't, so the physical sample curation cluster has been around probably for, I am horrified to say it out loud, but I think like 10 years at this point, maybe 8 years, but it's been around for a while. The goal is to support physical sample collections. So core repositories, museum collections, collections of rocks that are in random researcher closets. We want to support the sharing of data and metadata related to those physical samples so that people can find them, reuse them. And also a big thread that's kind of run through a lot of our work is helping sample collection curators show the impact of their collections and of their curatorial work. So that's been a big focus of the group. Some recent work, a big effort over the last couple of years has been developing this sample curation, scientific author guide for publishing research using physical samples, aka citation guidelines for researchers. We want to make it we want to make it easier for researchers to cite samples in their publications so that collections can better see what's being done with their collections, and so that we can better create that network of data resources related to sample collection. So we published a paper on that last year. We published the final version of our guidelines last year. And early this year, these guidelines were endorsed by ESIP's 140 plus partners. And so these are now official ESIP guidelines. And so this is a really big deal for us because I think it was like 5 years of work trying to pull these together. And thanks to everybody in this room who contributed to that work. Now we're kind of at a
crossroads.
Yeah. Yes. And Natalie and I are the co-chairs of this cluster. Probably should have mentioned that. Where we're trying to figure out what our next project is, and also is there still a critical mass of people that are interested in this problem area for the cluster to continue? There's not a lot of people here. I appreciate all of you. And part of that, I think, is, a lot of our collaborators are no longer working at the places that they used to work at, thanks to Doge. We also, I think, need to figure out if we are going to take on another project similar to this, like the develop of new guidelines. So that's our goal today, is to start talking through some of these other problems beyond just getting samples cited. And that's what we're calling the advanced topics in sample metadata curation. One of the outcomes, or this is a figure from one of the paper that we published last year. We often refer to this as the rat's nest figure, because what this represents is the mess of obstacles to sharing metadata about samples and how they've been used in different research projects. All of the orange triangles represent people problems. So they're places where people have a hard time assigning identifiers to samples, have a hard time putting those identifiers in their publications, things like that. All of the red triangles are technological problems where we just don't have appropriate infrastructure or software or something like that to share the metadata that we want to be sharing. So today we're going to try to focus mostly on orange problems, particularly around minting identifiers for like child samples. But we're trying to focus on like more of these people problems today, thinking about guidelines that we might want to develop. Before I move on, any questions? Anything I should define and neglected to earlier?
I was going to ask how many people here are familiar with or have heard of persistent identifiers for samples, like IGSNs. Okay. Just was double check, making sure that we can cover our bases.
Okay.
I think that was the main thing.
Okay. And with that, I'm going to hand it over to Natalie. Yeah, I'll go back to our agenda. So we have 2 talks, Natalie and then Joshua, and then we're going to break into some breakout sessions based on those talks and do some report outs. And also, I would love to have discussion about like future directions for the group as a whole for folks that are hoping to stay involved going forward. So, yeah, great. I'm going to hand it to Natalie.
Okay, so I wanted to talk today about a new NSF-funded grant that Andrea and I are leading together. And the reason that I wanted to bring it to ESIP is that this grant, in its DNA, is very community focused. And so I want to talk through kind of the overall goals of the grant and who we're working with, because I think there's a lot of folks at ESIP that hold a lot of different pieces here. And if you kind of keep that rat's nest diagrammed in your head, what we're trying to do in this grant in a nutshell is address some very targeted aspects of that diagram. So some very specific orange or red triangles. So this grant started in January. It's a 3-year grant co-funded by Geosciences Directorate and SAIS OAC. And I have up here the alt title is, the alt title is covered. So I have an IGSN, now what? And so we are working with 4 major stakeholders in that rat's nest figure: researchers, scholarly publishers, data repositories, and sample repositories, and doing some targeted work to move the needle and try to mitigate some of those challenges in the rat's nest diagram forward. And so this is our project team. We're Arizona based in terms of the leadership of the grant. So we have Andrea's student, Mac Borazon, is a graduate research assistant, and we'll have another one joining this fall. We also have a community advisory panel engaged as part of this work. And so we really wanted to make sure that we had representation from as many angles and sides of that rat's nest as possible to make sure that we're getting feedback all the way through the grant. So we meet with this panel quarterly, and we have representatives from DataCite, ARC Alliance for the kind of persistent identifier side of the infrastructure, a variety of national labs, and other academic spaces. So we get a range of different types of sample challenges in different contexts, as well as representation from the IGSN EV and scholarly publishers. So we're really grateful to our panel for their kind of perspectives and feedback. So for researchers, what we're focused on, I think in the grant we called it kind of professionalizing sample stewardship workflows. So getting researchers information and education about some basic best practices in sample management and sample identifier management. And the cornerstone of this work is a 4-semester fair sample stewardship training program. So we have stipend support for 37 researchers to engage with us in a 4-semester education and working program where they will receive lectures and kind of guest speakers from across the sample ecosystem, and then take that information and apply it in their own context to start to curate their sample collections. And then it's going to be reciprocal and iterative. So one of the major outcomes from that program will be the co-development of undergraduate and graduate education modules. So things that are going to be made publicly available that other folks that are not in the program could take in the future and implement in intro graduate courses, or take to their lab group for a meeting and talk through some of these kind of workflow changes. So I think, I think there might be folks in the room that know people or would be interested in that. So I wanted to kind of highlight that as something that's coming down the pipeline. The applications for that will open later in the fall, and we'll be posting on ESIP's Slack channels to kind of announce that. So we appreciate kind of circulating that. Sample repositories, do we have any folks that are managed collections? Curation, kind of? Yes, yeah, a couple. For that work, we're really looking at what are the kind of existing workflows and kind of scale of challenges that sample repositories deal with in their day-to-day as it relates to sample identifiers and metadata management. And so one piece of this is and I should say the light green items are items that are in progress. So we're developing and sending out a national landscape survey to about 300 US-based Earth and Space Science repositories, asking them questions about their challenges, their awareness of sample PIDs, kind of what they know about sample PIDs, if they do know about sample PIDs, and then some other kind of background information about their collections. This is going to be followed up with a couple select semi-structured interviews. So we're employing a lot of qualitative methods throughout this grant to kind of really get at some of those social problems and how they're interacting, again, in that rat's nest diagram with the technical. The other thing we're doing is we have 3 to 4 site visit case studies where we're going to go to the sites. We're starting with our first one this week, and we will be having a kind of structured walkthrough. Show us your curation workflows. Show us how you give out a loan. Show us what happens when a sample comes in, and looking very closely at what types of digital infrastructure and tools curators are using, where there's gaps, and how this intersects with persistent identifier infrastructure. Data repositories are, of course, an important community here this week. We will be running in collaboration with ESIP a webinar series on sample persistent identifier integration. So highlighting data repositories that are currently supporting sample persistent identifiers. This is a huge gap in that rat's nest figure, because it's one thing if we can get IGSNs or ARC identifiers minted for samples, but if they're not used and integrated and embedded in data site metadata for data sets, or they're not being picked up by indexers from other resources, we have some broken communication channels. So we're going to be working on some education there and supporting repositories and understanding that landscape and the values of supporting sample PIDs. We'll also be running hybrid format hackathons. And so at ESIP next year, we hope to have a co-located hackathon to help repositories start to actually implement some of this technology. And then finally, with scholarly publishers, there's kind of, this is one of the most underdeveloped spaces in that rat's nest diagram. And so we're doing some initial analysis to look at where persistent identifiers and identifiers in general are showing up in scholarly publications, and what the implications are for how we get publishers to better support sample persistent identifiers, and how we communicate that to researchers who are the authors of these publications. And so to that end, we'll also be doing some focus groups with editors and publishers. I wanted to highlight as well that we, as part of this, we've opened up a special collection co-hosted by 2 AGU journals, Earth and Space Science and JGR Planets. This is broad by design, and so we want to engage sample users, so people that have stories to share about the benefits of using and reusing samples and the benefits of leveraging sample collections, as well as the curators of those sample collections, talking about their collections, highlighting the science that those collections have enabled, talking about their workflows and challenges. And so this special collection will be open for submissions through 2028. Oops. And then I think this might be my last slide. This is just kind of a reiteration of a call to action, help us tame the rat's nest. And so I think that's part of what we're already doing here today in the context of the physical sample curation cluster. But for data repositories, or basically all of the initiatives that I just talked about, keep an eye out on ESIP general channels and the physical sample curation cluster Slack channel and listserv, and we'll have some calls for participation there. Let's see. Yeah, that is that. And happy to take any quick questions before we flip over to Joshua.
So thanks, that was great talk. You talked about the linkages between samples and data sets. That's obviously a great thing. I think like the relationship between data sets and files, that can be a one to many, 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 many problem.
Yeah.
Like, how do you recommend people, if you have a environmental data set that has 200,000 samples, how do you link that into a data set record?
Yeah. So guidelines for repositories was one potential avenue for this cluster that we talked about in the immediate aftermath of publishing this researcher author-focused guidelines. Right now, it is really manual. And I think that there's interesting things happening in the RDA space with the research citation objects that may help this. But it is a huge problem because you're also asking then data set contributors to manually put in all these PIDs.
Right. I guess I'll just turn that around and say that I think that obviously you can just list them, but then you end up with this really unwieldy metadata record that has possibly a landing page that's 6 miles long.
Yeah.
You know, that's the bigger problem. And so how do we build systems that ingest those kinds of quantities of samples is more of the issue than like just the linkage strikes me as not the problem. It's like how building systems around things that have one data set has many, 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 and possibly flipping it the other way around where samples reference the data sets to which they belong is a more natural linkage, because usually samples are part of a data set, whereas a data set has many samples.
Yeah, and so this is the exact sort of thing that we want to talk through when we have our hackathon. So if Data One is interested in participating, please stay in touch. So there has been work out of the complex citation working groups here and at RDA talking about this. And Dave Viegleis actually developed a little prototype, or at least he diagrammed out how he thought it would work with iSamples, maybe as an infrastructure. But my answer is that I don't have an answer, unfortunately. I think there's a bunch of different options, and we've talked about them in various fora for a couple years now. We need to now just try some and see what happens. I do think that there's a need for this is on the red triangle, that there's some sort of technical infrastructure piece that we are missing, if not several pieces of, unpacking files, having a consistent way of formatting those files. I think in Dave's version, it was just a list. Just list out the identifiers, and that should be enough. But where you put those files containing the identifiers is still an open question, and especially with the existing scholarly infrastructure, where it's like sometimes it's in supplementary materials, sometimes it's in So this is the exact sort of thing that we would love to try to make headway on for this project. We don't think we're going to magically fix it in 3 years with no development money, but I think, and that's this idea of like creating roadmaps. I think we can start outlining feasibilities for future work. So,
but even in early conversations, just in the course of kind of putting some of this together in a proposal, I think we were maybe not surprised, but it was interesting to talk to some data repository folks and realize that there's still some very primary gaps about, well, we support IGSNs. It's like, okay, you go look at where it is, and it's actually not getting in the DOI metadata. So I think it is like a learning by doing, and I think the hackathons will be illuminating.
I'm Ted Haberman from Metadata Game Changers. I think that's I think an interesting step in the direction towards what Matt was just asking about. You know, would be sort of like a list of use cases and what you want. Yeah. I mean, are you looking for someone searching for a sample for an IGSN and finding all of the data sets that it's in? And maybe samples can be in more than one data set. I don't know, but if what are the kinds of use cases that we're thinking about for those 200,000 samples? I mean, put them in a CSV and put it on Zenodo. I guess that's my answer. It's pretty straightforward.
Yeah, and actually, honestly, if there's interest and we have some data repository type focus, that could be a breakout today, even to start that. I think from the researcher side, or the way that we see it manifest on the other side of that is researcher wants to be able to go to an IGSN sample record and see all of the data and publications that are produced by it. So we need kind of that auto updating to work, and to get the auto updating to work, we need the data repositories to be able to take that information in. Yeah. So we're really fortunate today to hear from Joshua Bergback, who is at Oak Ridge National Lab and other collaborators here, talking about a specific use case for some of these advanced topics in PIDs and sample curation. So thanks so much, Joshua.
Thank you for the introduction, Natalie, Andrea. I appreciate getting to come here and talk about this. This has been a little bit of a whirlwind of a dive. I'm not a data scientist. My training is more botany and mycology, and now I find myself cosplaying as an ecosystem ecologist, as a technical professional running field campaigns and laboratory operations. But a need that was present in the project is one that very much had to do with physical samples, repositories, how we track them and how we organize them, as we'll demonstrate you. So this is work that I've been working on. We had a great summer intern not this year, but the year before, named Ethan, who came and did a lot of the physical labor involved in this work. And then Tom Ruggles and Terry Veliket, Terry's here, are our data management team for this project and have been incredibly helpful thinking it through. And then our PIs, Melanie and Dan. Does anyone know what this is? Any guesses? Give you a hint, it's a sample. It's not a stool sample. It is a piece of peat moss. So not moss, but this is the Catatown peat, so it's below the water table from a bog. It is from 50 to 75 centimeters below the surface. That's one bit of metadata. It's from plot 17, whatever that means. We have some coordinates for it and a date. So this is metadata. Super helpful, right? All right, this gives a little more context. Okay, so here's our 50 to 75 centimeter deep peat sample. It is from this hummock core. This is the structure in which it was collected. This is how it relates to the other samples that were collected at the same time in the same place. This is a lot of helpful metadata to put it into that broader context, right? So we have our different layers. We have the different segments. That's what it looked even worse when it was wet. That's what it looked like before. And then how it was handled. So there's now more metadata that we're putting our sample into context. We're also putting the campaign into context. How was it collected? How was it handled? How was it stored? How was it shipped? Here's a little more context on the location. And there you can see Terry processing the sample. This is plot 17. These are the coordinates where it was collected. We can even overlay exactly where that core was collected inside of plot 17, which is that turquoise circle. Now let's put the plot in context, and it is plot 17. It is a +9 Celsius ambient CO true whole ecosystem manipulation experimental plot. So the spruce experiment is, spruce stands for spruce and peatland response under changing environments. It's really great when you can make the first word of your acronym your acronym. Makes things really easy. So this is plot 17. This is the experiment. It's in northern Minnesota, about 45 minutes north of Grand Rapids. And this is a decade-long whole ecosystem manipulation experiment that is funded by the DOE. It is set up in a regression design where we have our unenclosed controls, we have our enclosed controls that have zero energy added, so zero heat energy, and then a series of plots at different temperatures above whatever the ambient temperature is. So the +9 is whatever the temperature, whether it's -40°C or 40°C. Well, probably never quite gets that hot up there, but definitely I have done field work in -30°C up here. So whatever it is, it's 9 degrees above that throughout for an entire decade, above and below ground, down to 2 meters into the peat. So that's why that peat sample is so special. It has been at 9 degrees warmer than its paired samples for quite a while now. Now putting the sample date into context. So every one of these metadata bits has a little bit more context, right? So this is the collection event. It was 4 days long. There were about 25 participants. We took 62-meter cores, manually, physically digging into the ground, pulling them up. Terry can attest it was hot and it was not easy. It took a long time, which is kind of fun. The kind of fun you have afterward when you're looking back on it, going, that was a good time. Believe me, I wish we would have gotten pictures of us after the week of doing all the peat processing. We were a mess. But it was 696 samples. time and temperature sensitive. Different people in these groups were doing different analyses. We pull the core out, and then it's mine and Terry's. It was a whole team of about 8 of us who were essentially just responsible for getting the peat out of the bog. Then we pass it down to this assembly line of researchers who take subsamples of that peat sample. Typically, and these are just the researchers that need fresh peat samples because they're being flash frozen or used for very quick chemical reactions or microbial responses, right? Other people get the samples after we get them back to Oak Ridge, and we can dry them and grind them if they're not doing temperature sensitive analyses. So these were the collaborators. We had government agencies like Forest Service, EPA, we had universities from as close as the University of Minnesota, just kind of a couple hours away, to people from the University of Zurich that were coming here to participate in this campaign, as well as a whole team of us from Oak Ridge, primarily a lot of technical staff who are not necessarily the researchers, but the people the researchers hired to do the really dirty jobs, like registering samples, because that is, turns out, complicated. All right, so here's a little bit more of then that date into context. So that was 2025. 2025 was the last year that the spruce experiment was running. Like I mentioned, it was a decadal experiment. So all these peat cores that we collected were the cumulative 10-year response of these temperature and CO2 treatments. So they're especially sort of special in the context of the experiment. It is a very large experiment with hundreds of researchers, nationally, internationally, hundreds of data sets, and it'll end up with a lot more publications, models, etc. And one thing that's sort of come up through this is a lot of the way this project has been ran has been that, it is DOE funded, it is publicly funded. We think of it as something that does belong to the people, and Tom and Terry have used terms like the scientific commons that is the spruce experiment. We invite people to come do research in our experiment because we can't do everything. We want these teams of other people coming in to do all kinds of analyses, bring their own ideas into the project, make their own data sets, contribute to the project. Much more than we would be able to on our own. And so part of the commons of that is the data, but also the samples, because we keep and archive samples from most of these sort of repositories. So here's what I wish my life looked like. I wish we went out and we collected samples, we analyzed them in the lab, we published a data set, and then maybe published a paper or two off of that data. This is a zoom in on one peat campaign, a small one in June 2015. And we can track from the actual peat coring samples themselves to the different lab groups at different institutions indicated by the little people heads, and then different analyses that are performed on them that go into various data sets that are then hosted in different data locations. So we're crossing, I think one of the most common things for us to cross across is IMSL and JGI, Joint Genome Institute. A lot of metabolomic, metagenomic, any sort of molecular microbial data. That's a whole nother repository than I typically work with. But between that and ESS Dive are probably our main repositories. But this is the situation right now, and it's about to get way more complicated because a bunch of data sets are going to start coming out that have cumulative 10-year responses, right? Where we're revisiting a few of these data sets that got published early, but now are including data sets all through the length of the experiment. So if we zoom out from one collection event to all of the collection events as of a couple years ago, this is what we get. Our green dots indicate the actual collection of events, which are the physical samples, the samples in the collection, and how they were analyzed. The gray represents the data sets. You're just indicated by numbers, but to show the connections between where these individual samples are cited in different data sets. And then blue represents, this is just like the lab groups of the researchers that were relevant to the peat data. And like I said, this is a simpler picture than we're about to have because we've got all these 10-year ones. I'm working on them right now, and it's a pain. But it's wonderful. It's very cool and interesting, but it gets a little daunting. How are we going to organize all of this? How are we going to make sure we maintain these connections and also have a physical archive that we can literally use too? So this is where PIDs come in. And I was so glad to find the ESIP guidance document for researchers on how to register samples, because that's why I'm here right now, is that this problem was recognized by project leadership. And then Terry and Melanie and a few people, I think, were aware of that sort of and went, man, it'd be great if someone would try to figure this out. And we could figure out how to implement this into our workflows. So that's what the idea is. We can take this sample, we've got our original sample here, and we can link it to the 4 data sets it's currently cited in, right? Perfect. We've got a PID. It's globally unique. Love it. Then we also really want to make sure that we can link the other samples from plot 17 across the different years, right? Because not only do we have 50 to 75 centimeters in 2015, but what was going on in 50 to 75 centimeters every single year that we've done sampling, which is just about every year the experiment has been running, right? We want to be able to link all those samples together. And then once we can do that, we essentially end up with one giant sort of data ecosystem, right? It's all related to each other. We can link the samples, we can link their treatments, we link all of their responses together, we see the data. I imagine people here are not the people I need to convince how cool that will be, right? Y'all are already thinking about this, that's why we're here. But that's just the peat samples. We have trees also, because the ecosystem also extends above ground, not just below ground. This is just kind of looking through some of our research, some of our internal documents for me to try to figure out all the data we have on specific trees. Literally like plot 17, tree 21, like we have potentially all of this data spanning a decade. And some of this is on a, like 5-minute interval. Some of this is collected twice ever in the history of the experiment, but it all links back to that tree. So how should I link all that together? Does the tree itself get a PID as a thing that sort of like a core, like a virtual core? Well, this is a virtual tree that we have cut into 1 meter segments and then done measurements on every single meter of it. Is that the correct way to do this? Should we do it on a plot level? Should we do it by collection event? What we're doing practically is we're lumping them together by collection event. I don't know if that's the best thing to do, but it's practically what we can do at this point in the experiment retrospectively. But what are we going to do next time is more what I'm most interested in. Or if someone can fix the problem for me right now, that would be great too. So I think you can kind of see where this is going. We have vegetation mega data set environments. We have environmental data sets. We have peat data sets. We have model inputs. We have model outputs. We have, and again, these are just the ones I could come up with off the top of my head going through some of our documentation. I think there's more to this. But if we can link it all together, Spruce is just one big data. It's just one data set. And that would be so cool. And I'm so excited to hopefully find a way to kind of get there. Or at least to use this as a lessons learned to know what to do next time. So say that's where we are right now. In progress, we're creating an internal inventory just so that we know what we have where, because we have dozens of lab group, not dozens, but there's probably been 7, 8 different PIs at ORNL that have managed different parts of this project. They didn't always talk to each other. Somebody has a freezer over there. Someone has a freezer down there. They have the same samples in 2 places, which is great sample stewardship in case one of the freezers dies, but nobody knows what has what. I've spent a lot of time just going through freezers, digging through, trying to find things. We have started registering and assigning them in IGSNs and CSAR. We've started with the peat cores as a pretty complex use case, but semi-manageable. Maybe actually, I thought it was the most complex case until I started thinking about the trees, and I think those are actually going to be harder. But so it was a pretty difficult use case that hopefully we can see if we can work our way through. We are publishing sample metadata sets and then updating all of the data sets that currently exist with the IGSNs that we've retroactively registered. And then hopefully we could at least demonstrate that sort of linkage within the peat data sets. Then if we can go from there up to the next step of how to link the plots, the trees, the below ground, other below ground things, that'll be pretty cool if we can figure it out. So we're going to be able to make it happen because this is what we're dealing with in reality. That's in the top left, that's a sample archive room that I inherited that has projects going back 30 years. Some of them are relevant to our current Spruce project. Some of them are relevant to experiments that ended 20, 30 years ago. So I've got to find people who haven't retired yet to try to dig through here to help me figure out what these things are. Do we need to keep them? How do I decode your code? What does Jesus moss mean? Thankfully, Dave Weston is still there, and they were digging up moss polystichum in the bog, and they could basically track it so far down. And with how slow the bog grows, they were actually kind of measuring, thinking this probably was around a couple thousand years ago. It might actually be that old. And then freezer is my least favorite thing ever, obviously. All right. So we have a sample archive, which is amazing. We got resources to create a permanent archive where we can store these things. That is lovely. Not everyone always gets to have that. We've never had it before. We've had disorganized rooms like that. So thankfully, we've got freezers, we've got dry storage, we've got -80s, with the specific goal to preserve and store these samples for as long as they are useful, not necessarily forever, which is somewhere where we're slightly different than maybe some other repositories. We want these samples to be used by the community because that is what they are for. They are not necessarily meant to be forever samples. I have thrown away people's lives worth of samples, and I feel really sad about it. But like when we did some clean outs and went, this box says it could be disposed of, July 3rd, 2000, and it had been there in the shelf the whole time. But I have dumped out huge amounts of samples. We've come up with a system to make sure that we can record what's in our inventory and some of the process knowledge we need to be able to keep, to be able to, one, make sure that we're maintaining metadata to make these samples useful, because if they're just a sample with a code and you don't know what it means, that's obviously not useful. And also, if there's any hazards, who's responsible for them? Who do we ask when we have questions? Who gets to make a call of whether we're going to subsample something off to somebody for analysis? Challenges, can we make this a one-size-fits-all solution? We have a lot of, I don't know. I guess everyone has complexity. We have complexity. Can we make it fit our system? I think maybe. We'll see. Overcoming research culture is still really hard, as much as I'm trying to top down. I'm actually trying to bottom up manage it. We're doing our very final campaigns. The experiment's over, but I'm trying to, implement this here at the very end. People have a decade or more of going up to the site, collecting samples, and just writing 2019 shrub stems, and that's your metadata now. Can we get people to not behave that way anymore? And can we really scale this up? Because, I don't know, we're getting there with the peat samples, but just say it's taken a lot longer than I wanted it to. I think it's worth it, but are we going to be able to scale this all the way up through all of Spruce, from 2 meters below ground to 900 cubic meters above? So thanks for the opportunity to share. Like I said, Ethan, bless his heart, created all the CISA registrations, got the IGSNs for everything, tracked down metadata from people who weren't even currently employed anymore, who were willing to answer questions about some of what we did. We're in the process of finalizing those for ESS Dive, but we have all the IGSNs, and then we'll update the data sets. And then Tom and Terry, Melanie, Dan, and Paul. Here is a link to our sample archive. Please, use our samples. I know you guys probably aren't actually the right one, but if you know people back at your institutions that work on projects that you're familiar with that might find this interesting, we have these samples that I think are really valuable. You can never redo a 10-year whole ecosystem manipulation experiment. And if somebody has a really cool way to analyze these peat cores to tell us about how the world works, that's exactly what we want. So use up our samples so I don't have to throw them away.
Hi, Erin Robinson, Metadata Game Changers. And I worked with Natalie and Andrea on some of the sample work, and work with another group in Maria on similar things, but and marine data, but this is really familiar. So what I was wondering is, could you talk about what kind of field information management is happening sort of at the time of taking the bog core, and just a little more about that.
It very much depends. When we're taking the cores, we are not really collecting that much field information. We are collecting the location of the plot it was collected. We were collecting temperatures, but they weren't analytical temperatures. We have below ground sensors to actually measure that. It was just sort of spot check the temperature treatments below ground. We are taking fresh weights. We are taking, which is in a bog. It's windy. please round off all the decimal points. I always tell people. I don't know, Terry, are we collecting that much more field data? I think the nice thing about working in such a defined plots with plot maps is that we have a lot of that data already built into the structure of how it's laid out and organized, so that when we do take a tree sample, all I got to do is write down what tree it is and then how high it was. And then depending on what, sometimes it's what side, what exposure is it on? Is it, so it so much varies between individual researchers and campaign to campaign. So that's a fun imagining formalizing that in the future. Oh, we have hummock or hollow too in the bog, obviously. We were also recording water table depth because the elevation has changed with the treatments, and then sometimes your plots are actually different absolute elevations, even though Originally there was proximity to trees was part of it too, but that was dropped because of effort it takes to excavate 2 meters of peat. You can do like close to trees and not close to trees, thankfully.
Brett Longworth from Hui. First of all, this is like giving me slight PSD for many of the projects that I've worked on. Secondly, so do you have bits of metadata that you found to be useful for figuring out samples later? Like any project where you're tired, where you have a bunch of people that aren't necessarily super experienced, you'll end up with a sample where you're like, where is this from? So your temperature thing reminded me of that, because then obviously it's not winter if it's 25C. But things like, or also the Jesus moss reminded me of that. Do you find that you have metadata that are especially useful for sorting things like that out?
Especially playing catch up after the fact in this kind of stressful world. And Terry is the one who's, I think, was really good about making sure I did this with mine before I was thinking about this as much, as defining your sample code structure super explicitly in your metadata. What does every single letter number, what is the order, what is the pattern, right? That is really helpful when people do that. And when it's achievable, you don't always get to know ahead of time. You can't plan ahead. You don't know. So I've tried to do that in ways where it didn't work out. With the peat coring, you kind of know how deep you're going to go, and it's relatively easy to lay that out systematically ahead of time. I think we have... It's kind of nice having being so structured, having these plots and the maps. I do wish the maps in the past had always indicated what year things were going. We haven't really been able to figure out where the cores were in past years because they were just marked with a C on the map, and that's a little annoying. We can technically go back every year and look at the archived versions of the plot maps and kind of figure out, but I'm not sure we're going to do anything with that data anyways. I don't, thankfully, even if we didn't have the structure set up from the start, most people are very good at recording their metadata. Again, it's very researcher specific. I have certain people I like a lot more than others. And, people who I ask them, what are these samples on this shelf here? And they'll go, well, let me email you a spreadsheet with all of that information. I go, thank you so much. And then I, talk to somebody else and, oh, I think I remember those. Let's see. It was fall, and you go, okay, great. Thanks. I don't know. It's really researcher culture, probably more than anything, that I count on some people versus others. And I think it's side plug, but that's why you put certain people in charge of certain things or not. You need to have these considerations before you do your sampling campaigns as much as possible, because it's a skill that some people have and excel at, and not that some other people don't.
I think we've learned a lot. Joshua implemented this in 2024, right? And then we did it in 2025 in the field as well. And so I know the first year we were trying to pre-label things, and we had 2 bags weren't the right size, and just trying to think about that. But I think the people, we have technicians on site at Spruce that are there about daily, and they're very on top of things, and so they are very good at helping. I know when they started all this, it was just like, what are we doing? I'm sure because everybody's doing field stuff, and it was in 2012 or 2010. I mean, this was a long time ago for data management stuff, and So we had a crew, and as Joshua said, Ethan was really, really good. It took 4 of us to figure out, with the help of Joan Damerow, to figure out what we were doing.
Yeah.
And so having consistent people and a plan in place has been really good. And pre-printing the QR codes, getting the labels set up, registering those, and having those in the field with us. So when we were chopping things up and putting them in bags, they already had the labels on them. And then we pass those off. Once they get passed, they're off to these other researchers, and we're not in control of them unless, they're not under our funding anymore. So
we did pre-print a bunch of labels so that, and ask them to use them and to reference the IGSN and the sample code on there. And they had live barcodes, so they couldn't complain about not being able to find the metadata, right? So we did try to encourage that. Another nice thing we have in our project is that Tom and Terry are also the data management gatekeepers. So when someone is going to submit a spruce data set, and if they didn't include them, we can kind of jump in and get them in there before they make it to the data repositories.
So this is a transition towards breakout question. Can you go back to that slide that you showed with the network of all of the samples and data products and things like that? Yeah, perfect. So, and this is also building off of what Terry was just talking about. Can you talk through your PID assignment workflow? So are you always pre-registering IGSNs and then giving them to people? Are you minting IGSNs when stuff gets back to the lab? Are you and also are you minting IGSNs for all of the children data or samples that are coming off of these things?
Perfect. Yeah, I probably should have talked a little more about the hierarchy. I did mention how we've decided to structure, for now at least, the hierarchy around the collection events, and that's nice because then it reflects the physical infrastructure of the archive, because I got a box of 2014 peat samples. But it's not worth it to go back and relabel every single hundreds and hundreds of tubes, right? But I can label the box. In 2024 was the first time we tried to field trial this with the peat coring campaign on a smaller scale than this one that I was showing you. This was the 2025 campaign. This was by the most complex thing we've been able to pre-register and do ahead of time. This worked relatively well pre-registering because we were pretty aware of what samples we're going to collect. We know how low we're going to go, more or less. The decision of whether to do that or not mainly has to do with how predictable the campaign is. This one's relatively predictable. Did I mint some ghost PIDs that didn't actually get samples because we hit mineral soil before we hit 175? I did mint a few. Not very many, though, and we can deactivate those. Not the end of the world. Thankfully, I did not pre-register them, but I tried to pre-label one of our tree campaigns. And then it went to the records of, okay, well, on the paper, it said that the tree was only 4 meters tall, but when we actually cut it down, it was 4.7. And now I have a whole nother 1 meter increment that I need labels for that I didn't print, because our records, once you actually just cut it down and lay it down, it's a little different than when you're out there doing a height poll. Some of the tree campaigns I would not pre-register just because of the complexity of not knowing what you're going to run into when you run into nature. Same with the shrub sampling campaigns. We have not gone down the route. So all of our parent-child structure gets down to the sample that comes out of the spruce enclosure. We've not minted PIDs for your lab group gets this PID, your lab group gets that PID. When I send this out to you, I'm going to get a PID for you. We just haven't done that. It might be something we want to do maybe in the next iteration, but it's playing so far catch up that's we're just trying to get one thing for the thing that actually came out of nature. And I think that's probably the most useful for our use case, because your piece of my homogenized peat sample isn't really different than my piece of my homogenized peat sample, or it's not really supposed to be. Now, there could be sample preservation metadata that would be different, so there may be reasons you might want to make sure that this one was collected flash frozen. DNA is great. Like RNA, you could use this for microbial stuff, which would definitely be different from the ones that I dry in an oven and grind up. So there's something to think about. Did that answer?
Yeah, that was great. I think what I was trying to get at is there are open questions about what are I think maybe it might actually be the slide before this. Yeah, I I don't know. Yeah. So what is the unit to which an IGSN should be assigned? And I think we have conversations about this at CSAR, but that's also a conversation that the IGSN community in general has been having, and people have differing opinions. And I'm relatively agnostic about it, but it was helpful to hear how you guys are doing it. So it sounds like one identifier for one entity, and then you are not doing children identifiers right now.
So each one of these, right now, each one of these cores is in there as a virtual core.
Got it.
And there's complications because, we take our cores and we cut them into these sections and we homogenize them and then we give them to people to subsample out of. Well, a few teams are different and they don't use those exact segments. They use their own segments. So if they want to cite the core, they need a PID for the whole core, which still does tie to our subsamples from that core, but they're only using like a homogenized piece of a certain range of this. So so each core gets a PID, and then each of these sections in the core gets a PID. Because everybody's special.
Right. Any last questions before we move on? Yeah.
So where do those PIDs go?
So right now they are going into data sets in ESS Dive. So because of the complexity of linkage between data sets and sample sets, right? In ESS Dive, there's options to include your sample metadata in a data set, which makes a lot of sense if you have this kind of system. You can just embed your sample metadata in there because it's not tied to 10 data sets, right? So instead, what we're doing is registering each sample set as a data set with no analytical results, just with the sample metadata. And that makes it citable, right? Because it is an object in ESS Dive. It is a data set. So you can cite the sample collection events that you used. And those include the metadata and methods on how they were collected. So you don't have to also regurgitate all of that.
And so do those become data site DOIs at some point?
They are DOIs. Okay. Terry's more of the data.
In ESS Dive, that's the repository they're in?
Yes. Yeah. It's like,
is that it? Data one? Or is it a self-managed thing that's got something at Data one?
So it's run by the lab.
Yeah.
And Dr. There, they run requests. They use our software and part of Data One, and all that stuff shows up in Data One search. But the data sets are managed and stored on the system.
Thanks.
And I think Neon is doing a similar approach with the data sets of just the samples as well. I don't know that they meant DOIs. Yeah. Okay.
I would love to talk to some of those Neon people. So if we know who they are, let me know.
So each IGSN is a DOI that comes out of data site. So each one of the samples, I don't know how many were there.
600.
120, 300. I can't remember how many we did. And those are all for like the 2025 campaign. Each one of those samples got an IGSN. So they all have DOIs. And then that data set that Joshua mentioned, it has a citable DOI. So that goes back to the question, you can cite the whole package with one DOI. And if you're using multiple samples out of there, you pull the IGSNs out and put them in your data set. But you can also just reference that whole data set because it has all the collection information in there. This is where the cross-referencing. But in ESS Dive, which is the DOE repository that this project is supposed to put their data, they don't have a good place for us currently to put those IGSNs, which is what we were talking about, just a list. And since the data set that we made is all the IGSN, all those are listed in a table in a spreadsheet or tabular. But we're still back and forth as to should they be listed in like the methods section individually, or should they just be in the table? Because ESS Dive has got this way to search now if you're using APIs and AI prompts and LLMs, and you can search inside data sets. So you should be able to find the IGSN number.
Should
So we've been having lots of conversations with the ESS Dive team about just how we represent all this stuff. Like this, I think this is one of the motivating cases for them about how to extend the software, the data repository infrastructure to support this. And the challenge is that any given quote data set in ESS Dive might have hundreds or thousands or tens of thousands of DOIs associated with it, all through this complex linkage to this experimental design, which is playing out over decades of research, right? So it's like, doesn't all happen at once, it changes through. And so that's the challenge. And then the DOIs are issued from DataSite, but that comes through OSTI. And then that in turn is actually mediated by our software Metacat. So when you go to mint DOIs, you put in dataset metadata that then triggers a push to OSTI for all that metadata, and then OSTI pushes to DataSite to actually get the DataSite metadata there. So there's actually a complex chain of software involved in trying to figure out who can extract what from where and get it to the right places. That's why I asked this question about how do you list so many samples on a dataset record when it's like, it's a very complex set of relationships. And we've been finding that like OSTI drops some of the stuff in the translation to DataSite as well. So trying to figure out who's responsible for what and how these linkages occur in a way that everybody can predict it into the future so that people writing software systems on it know where to look for stuff is a pretty hard problem because it's a complex set of information that's, yeah.
So just OSTI DOIs?
Yeah, OSTI is like the identifier registration service for the Department of Energy. Yes, yeah, sorry.
Yes.
No, that's also there's a bunch of history at ESS Dive. So there are some projects that have their own DOI prefixes that have been around for a long time, like the CDX stuff and whatnot. And there's newer stuff that is owned by ESS Dive through OSTI. And then there are other projects as well. So I think there's no one answer to that, like who owns the DOIs for these data sets.
So when we envisioned this session and talked with the cluster, we all take time to be here in person this week. We really wanted to give space for conversations that emerge and kind of work on topics that are of interest to the folks that are actually in the room today. So we had seeded some possible ideas. I guess we've kind of been talking about two, but we've been maybe more talking about it in kind of some of the more technical linkage and the data set and large sample problems, which I think would be a fantastic breakout group. But I wanted to basically, I guess, open it up if there's any burning topics or kind of interest that we haven't yet talked about or surfaced in the room that people are interested in talking about in the next. We'll break out for about 20 minutes. And then come back together, and maybe we have two groups or so. I think it would be maybe if it's of interest, like it'd be interesting to have a group diagram some of these connections that we've been talking about and the current points of failure, and the current needs of like at this stage with this entity, we can't pass large sets of samples, or this information is getting dropped, or further upstream or downstream, the way you think of it. What are the kind of just practical limitations in the lab, in the curation space? But other topics.
Yeah, anything else you'd like to talk about in a smaller group for the next 20 minutes?
I'll just put a quick plug I was going to make during the talk, and I realized I forgot, was on Terry's poster about data set authorship and how to handle authorship issues when we've had complete turnover in staff, maybe from the 2009 samples to now. When from a usability standpoint, you want it all in one data set. How do you determine that? What order are they supposed to be in? Just I'm going to talk about that from the poster, and I guess it relates to that.
With number one, yeah, and we've had some of the State Geological Survey cluster participants have that same challenge, where it's these longer term data sets that get updated. So maybe 1 and 2.
Sure.
No, but we were going to use it as a seed. No, but we were going to use it as a seed as like kind of the main thing that's being used in the publication space, and then point out all the places that it fails for samples and what do we need to do about that. And kind of where for some of these, that I think the downstream thing there is like, is there a place for the IGSN EB to, start to have some recommendations on metadata fields. Yeah. And what are those metadata fields that are pertinent to the sample use case overall?
I mean, I'll tell you, we've been throwing around using a modified credit taxonomy just because we don't have anything else.
Yeah. That's where we're coming from with that. Yeah. So let's do those two.
Yeah. Yeah. Credit just is, I have some experience in credit data sets. Actually, is I think that credit, in my own personal opinion, so I can be opinionated. Starting with credit, it's really there's no reason to do it unless we're
right. Well, you can start from.
We can start from We can start from somewhere else.
You can start from throwing out credit and working towards something new.
Yeah. Okay. No, we don't think we're gonna run with credit. Yeah. So I think maybe those are our two groups then. I'll make an executive call. So let's have a discussion about because I think there were some really interesting connections and conversation there talking about ESS Dive and the linkages with Data One and OSTI. And feel free to use the board. We did have in the session notes breakout. We had 3 breakout group locations at the lower part if you want to use those notes. But feel free to do. It sounded like a diagramming thing to me, but go with that. And then on this side of the room, we'll have everyone who wants to break the credit taxonomy and talk about the roles and kind of metadata that are needed for samples, specifically, and the different people involved in that. So everybody can get up and move to wherever you want to be. We will reconvene at 3:25 and do very short report back. So nominate someone, tell someone that they're going to be their report back. Okay. It's report out time. Who wants to talk from the anti-credit group? Anti-credit.
Attention, please, from our group. We didn't come to any conclusions except for that accreditation and authorship is a challenge, and it's very. Thank you. Yes, and it could be. It's a kind of sample of choice. Ammonite was what I was thinking, and we have a shovel. There are many people associated with a physical sample. It's very disciplinary specific. We've extricated role from the role a person has on a project is unique from the action that they have related to the sample. So we're thinking to add a metadata field that has roles that are discipline agnostic to CSAR and see if people hate it and then change it based on that. I think that's what we got to.
Well, and then I was going to say the takeaway from, this is hard and we don't really know what to do when we didn't fix it was maybe we try to turn this into a project for the cluster over a hopefully not super long time period.
We talked about that it would be so the when we were trying to understand each other, and then we kind of got the core issue is what are the roles and how do you define them, and it is something that's potentially tractable for the cluster to make some what is the generalizable core here of the pertinent roles, and then talking about in fact they often I think tie to the site, so and then talking about how that gets embedded in the IGSN metadata and how you support it in a system like CSAR when you have field campaigns and kind of concepts of like cover sheets of listing out who was involved, et cetera.
Okay, so this group was talking about the relationship between samples and child samples. And we talked about the relationships between different kinds of PID species. And so thinking about how IGSNs and DOIs relate to other things like UUIDs and ARCs, and some experiments that we might try. And then that's all I've got here. Anybody want to add?
One of the biggest barriers is cost. If your DOIs cost $5 a DOI and you have 5 billion of them, are you going to mint any?
Yeah,
you know?
I would add.
Yeah.
So I would modify on that. So their DOIs aren't cost prohibitive, but the way that they're structured, I think right now is just making us think that if we have, 1 billion samples in a museum, for instance, that maybe it's not even necessary to have a DOI for everything. Cost is one part of it, but not the entire, that's not the only reason we might consider a different kind of identifier for that extended large tail of things. Yeah.
Nice. Yeah.
Sorry.
And an addendum to that I thought was interesting that came up was that the essence of an IGSN isn't in the form of the identifier, it's in the form of the metadata. So we could adopt the same IGSN sample metadata profile and use that with ARCs or with UUIDs or other identifier form strings. It doesn't, that's really about the metadata profile, not the form of the syntax of the identifier string.
Yeah.
Great.
Yeah, so we talked about getting John Coonsey to experiment with using IGSN metadata, data site metadata with ARCs. But one thing that the cluster might want to discuss further is what are the use cases for an IGSN versus an ARC? And can we help, or some other permanent identifier? And I think something we want to talk more about is why do you need a citable identifier versus just a unique identifier? And that's something that came up when we were writing the guidelines. Do you really need an IGSN for a water sample that's going to get destroyed in analysis? And I think that's a really reasonable thing to talk through that we should maybe converse more about in the future. So, yeah.
Yeah, I think what Matt just said is really important, that the content of the metadata, but this question about costs of DOIs, there's really, I'm dealing with this with RRIDs now, and there's really a lot that you lose. So if you're using DataCite effectively and you're using a DataCite DOI, there's a lot of things that you gain for that. It's more like 5 cents than $5, but you're losing a lot.
Yeah, and I have my own gap on. I always kind of think about indexing and kind of where these things are getting picked up and the pros and cons of that. And I think that there's a lot kind of just laying out the differences there and the advantages and the disadvantages and why that feeds into that decision ultimately of IGSN versus ARC versus. Yeah. So maybe Andrea has a couple more sonnets to write to IGSN or not to IGSN, to ARC or not to ARC.
I'm going to rewrite all of Hamlet to exclusively be about PIDs.
Yeah.
It's going to be great.
Well, that's great. So there's some actionable things to take back to the cluster. I think Andrea had a slide here. I'll just put it up and then we can walk right out. The cluster will meet. We meet once a month. The next meeting is August 25th at 4 p.m. ET. We'll be doing a debrief of ESIP and having an open discussion about the future directions and priorities for the cluster. We are also looking for a co-chair or co-chairs. I will be stepping down from co-chairship. So if you're interested in that, come find us this week or message us on Slack and we will happily talk to you about that. So thank you all for attending and we'll stick around in case there's more discussion.